Yeah.
Is it recording? It should have started, right? Yes. Okay. So hi, everybody. Once again, it's a great pleasure to have Krasimira Tsaneva-Atanasova. I hope I didn't mangle the pronunciation too much here with us. Krasi is a professor at the University of Exeter and also a very senior person in administration. She did her PhD in the University of Auckland in New Zealand and then did postdocs at the illustrious NIDDK Research Unit in Nonlinear Dynamics and also at the NS in Paris, and then started a position initially in Bristol and then moved to Exeter, if I'm correct, where she's now a full professor. She'll talk to us today about saddle node Separatrix-Loops and neural network dynamics. So over to you, Krasi. So if there are any questions, please post them in the chat as usual, and I'll interrupt. And there will, of course, be time for questions at the end as well. Over to you.
Thank you very much. Thank you for inviting me. And please do feel free to interrupt me. Because I'm on my laptop, so I can't see anybody or anything, but feel free to interrupt me if you have questions as I go over my slides. So so the topic of my talk is saddle node Separatrix-Loop and neuronal network dynamics, and I hope I will. So I will kind of be able to to introduce this what I mean by saddle node Separatrix-Loops and how this connects to neuronal network dynamics. It is really a work that is, well, some of the work that I'm going to show is published, but some of it is not, and it's being kind of currently prepared for publication. So it is definitely something that we are working very actively at the moment on. Let's see. So okay. So I'd like to start with just making sure that we are all in the same page by introducing this notion of saddle node Separatrix-Loops. Dates back to like 80s when, well, first publication by Lukyanov in a Journal of Differential Equations, in Russian actually, from 1982, introduces this kind of phenomena, which involves a transition between homoclinic and heteroclinic. You'll see this a lot in my talk. It's also known as saddle node homoclinic orbit or saddle node loop bifurcation. In 87, Schechter, I'm not sure if I pronounced his name correctly, Stephen, publishes this paper in the same Journal of Mathematical Analysis, the saddle node Separatrix-Loop bifurcation, where I have taken these pictures from these original publications, where he basically unfolds this in two parameter. The main kind of phase portraits that I want to draw your attention to are these two. By the way, can you see my mouse moving around the screen?
Yes, we can. Yeah,
okay, good. So basically, before the transition or the kind of the bifurcation, we have a saddle node and the homoclinic, and as this transition occurs, this saddle node and this saddle here that's involved in the homoclinic collide, and a kind of saddle node on invariant circle or SNCC appears. So that's what this bifurcation is about. Like soon after that, in 90s, there were two publications independently published using slightly different machinery from dynamical systems that talk about this phenomena in more general. In the general case, they refer to higher dimensions than 2D, really. Fast forwarding to a publication by Izikevich in 2000, he mentions this phenomena in the context of applications. Here are again the phase portraits from this original publication, where he says this bifurcation, the saddle node Separatrix-Loop bifurcation, plays an important role in at least 4 types of bursting, including fault homoclinic, which is also known as square wave. But he also makes a remark that it could be found in models of Morris-Lecar, Wilson-Cowan type. And here is a is a paper actually that shows this in a model of bursting behavior, biophysical model that explains spontaneous bursting behavior in developing retina, published in 2019, where they basically, this is here in the two-parameter bifurcation diagram. This is the point where this bifurcation. Oh, sorry, the point is here. It's a small picture. It's where this homoclinic, which is given in kind of cyan, turns into a SNC. And here is, yeah, so Izikevich makes this remark. More recently, this phenomenon has been kind of implicated in the effect of temperature on action potential dynamics. This is a German group and paper from a couple of years ago in Nature Communications that actually, again, I have a quote here. The paper talks about phenomena that has received little attention so far, and it's implicated in action potential dynamics, basically switching to homoclinic action potentials and implications for neural encoding and synchronization. And here is this kind of schematic representation of this transition, where we have a saddle node and the saddle and the homoclinic. They kind of collide, and after the bifurcation, we have a SNC. And this also here in this paper, they do show this also using recordings from a single neuron, like patch clamp recordings, and they relate their model analysis to their recordings and the implications for encoding and synchronization. So in our work, that's like a bit of a history, we came across this bifurcation in modeling GABA glutamate neuronal networks in the posterodorsal medial amygdala. So I will now introduce, the motivation for our modeling and what we have found in this model and where we are taking this forward in the remaining part of my talk. First, a little bit about the actual system that we are looking at. The amygdala and the medial amygdala within the amygdala is a brain region that integrates environmental cues from different sensory pathways and signals to other parts of the brain, including hypothalamus. And this is the kind of the pathway that we are looking, the signaling pathway here, or the signal transduction in the brain that we are interested in. As there are more and more emerging evidences that this is the kind of this is the way stress mediates or modulates reproduction, or at least one of the ways in which effects of stress impact on the reproductive function. And this has been something that is of interest to one of my long-standing collaborators from King's College London, Kevin O'Byrne, who is a reproductive physiologist and has been studying the reproductive system as well as the effect of stress for many years. And we have been cooperating together for now almost 10 years on various aspects of the reproductive system. In this particular project, we're looking at the effect of stress mediated via the so-called posterodorsal medial amygdala, which is a subregion of the medial amygdala, or I will call it shortly, briefly, MEPD. And we have recruited a quite talented PhD student, Kate, who is now in her second year, well, at the end of her second year, and is been developing this modeling and analysis. So the work that I'm going to present to you is basically some experimental data from Kevin's lab and Kate's modeling and analysis just before I present the data, I'd like to give you a brief overview of the reproductive system. Many of you may be familiar with this, but some may not. The reproductive system is basically controlled or regulated by the so-called reproductive axis, commonly referred to as HPG, due to the three main components that organize this axis. And these are hypothalamus in the brain, well here, the pituitary gland, and there are specialized cells in the pituitary gland that respond to signals from the brain to release hormones that then travel through the bloodstream to the gonads, ovaries in females and testes in male, to kind of secrete reproductive hormones such as estrogen, progesterone, testosterone, that then in turn feed back into the hypothalamus and the pituitary gland. So it's a whole body physiological system. And I already mentioned there are some well-established messengers that are responsible for the communication between the hypothalamus, pituitary gland, and the gonads. And here I've listed some of them: GnRH, gonadotropin releasing hormone that is secreted by GnRH neurons in the hypothalamus. LH and FSH are the first hormones of reproduction secreted by the gonadotropes in the pituitary gland. And then we have the sex steroids here, these ones secreted by the gonads. And disruption of this system leads to hormone and fertility disorders. So given the importance of the reproductive system, it's of course of interest to try to understand better how the system is regulated and modulated. Over the years, for me personally, this is a system that I've been working for since basically almost 20 years in various aspects of it, starting with looking at that so-called non-monotonic frequency response relationship, the signals coming from the brain. The GnRH signal is highly dynamic, it's pulsatile, and for a long time it has been known that there is an optimal frequency of stimulation that results in the optimal release of LH and FSH from the gonadotropes. So we have been looking at modeling and analyzing these frequency response relationships for a while with collaborators in Bristol. More recently with Kevin, we have been looking at the driver of this pulsatile secretion. That is also, it's an upstream of the GnRH neurons nucleus in the hypothalamus called arcuate nucleus, where there are specialized kind of neuronal cells that express these type of neuropeptides, kisspeptin, neurokinin B, and dynorphin. So that's why they're known as Scandi neurons. So we have been modeling this neuronal network, which I'm not going to talk today about. It's been already published using experimental data. And more recently, the topic of today's talk, we have turned our attention to looking at the modulators of these dynamics, specifically the modulation by stress mediated via the MePD, that nucleus in the amygdala. So these are just some examples of the in vivo experimental observations that we started with, but I think they are sufficient to illustrate the modeling that we've done and the analysis that follows. So in Kevin's lab a couple of years ago, they were basically stimulating using optogenetics neuronal populations, selectively neuronal populations in the amygdala, in this case, populations that secrete kisspeptin, and then measuring, like you can see here the picture of the mouse, that's done all in vivo, in live, really behaving animals. Then taking blood from the tail of the mice and measuring the LH levels, the hormone levels. which are pulsatile, and so they can be quantified in terms of frequency. And so what they found experimentally is that stimulating these kisspeptin neurons in the amygdala have an effect on the LH frequency. And in particular, here it increases the frequency. And then in the presence of GABA receptor antagonists, this effect is opposite. Basically, the LH frequency is decreased. So this is the observations we started with, building a model, trying to explain how this might be happening. And to investigate this, so we propose this network model of the amygdala neuronal populations and couple it to our previously published model of the driver of pulsatile LH secretion. So this is schematic representation of the model. And the assumptions that we have made here, I'm not showing experimental evidence for that, but it is known from from other group studies that look at histology and gene expression markers, that there are glutamatergic and GABAergic populations of neurons in this brain region, as well as neurons that contain kisspeptin. So we basically consider relatively simple circuit where we have glutamate and two populations of GABA that are coupled via this GABA-GABA interaction. Again, there are evidences in the literature for the presence of such kind of circuit in the amygdala. And we make an assumption here that one of these populations of GABA projects down to the arcuate, or projects basically to the arcuate nucleus in the hypothalamus. And we also can't basically exclude that glutamate neuronal population also has projections to the arcuate. These projections are functionally basically identified. In Kevin's lab, they have done experiments where they stimulate selectively and optogenetically just the projection, just the kind of the projections into the arcuate, and they have looked at the effect on the LH pulse dynamics as well. Again, I'm not showing this here, but this is all published. So in the model, we have these three neuronal populations that are interacting, and kisspeptin is just an input.
Krasi,
Yes.
the second subpopulation of GABA, how
Yeah.
come you don't have inhibition from GABA to glutamate?
You mean from here to here?
Yes, exactly.
Yeah, so initially we started with everything. We had all of these, all possible connections. Basically, there was GABA and GABA inhibiting each other. And actually, we now, I'll show you at the, well, I'm not going to show everything, but now we have some recordings of these, and we know that there is this backward interaction probably as well, which is maybe not as strong as the So basically, we started with everything, but then we decided to simplify it in order to, basically, we found that we can ignore these other connections to be able to explain that specific kisspeptin data, but in reality, they're all there. And as I said, initially in the model, we had all of them, but then we set some of those connections.
So you've done like some sort of sensitivity analysis to see that they were contributing little to the, I see. Okay, thanks.
For this specific store that I'm showing. But as I say, I think there are other data now that we are collecting, and we think that in some of those experiments, these connections can, well, to explain some of those, these connections cannot be ignored. But yeah, so I probably should have, maybe on the next slide, see here we have everything, but we set some of those to 0, as I said. So that's the full circuit, including this self-excitation and self-inhibition. Some of them we had to keep, like for the glutamate, but some of them we could set to 0. I think these ones were set to 0, and this one here, and then this one here, and this one. No, this one is there. This one is not there. But yeah, in terms of initial model, we had everything, and then we tried to simplify when we performed bifurcation analysis and look at some of the dynamical kind of bifurcations and phenomena involved. So this is the full model, and it's a Wilson-Cowan model. For those of you who are not familiar, I'll briefly introduce it. Basically, what we have in this type of modeling, there is a kind of connectivity functions that are modeling the interactions between these neuronal populations, and they are basically they represent the weighted connectivity or the contribution of each of these that Anmar just asked me to the overall circuit interactions. And in our analysis, the modeling that I'm going to show you, we have ignored some of them for the moment. We also have that stimulus response function, which is a nonlinear type of Boltzmann function. And we have the three ordinary differential equations that represent the basically the behavior of the rate of change, or how these neuronal populations behave. So basically, these equations are linear in terms of this activity of those populations, and nonlinear in terms of how they interact, which is given here. Right. The way we implemented here the input from kisspeptin in order to compare to the experimental data is by adding this constant to this weighted connectivity functions, and kind of scaling it by this alpha. Basically, we, instead of having two different inputs to this, that's how the original actually Wilson-Cowan works. But again, in our endeavor to minimize number of parameters, we decided to basically represent these inputs as using just one parameter alpha. And alpha is the proportion of kisspeptin that stimulates here the glutamatergic population, and 1 minus alpha weights the input that goes to the GABAergic population, one of them. Again, we're making here an assumption that kisspeptin stimulates only those two, doesn't directly affect this second GABAergic population. So now, starting with this experimental observation, stimulating this kisspeptin neurons, and basically varying this parameter in the model, Kp, and looking at what the effect on the LH frequency might be. Not directly here in what I'm going to show you. It's a bifurcation diagram, but we can later. I'll show you that it actually works. But basically, what we are looking is suppressing the. I need to go back to be able to explain it properly. Sorry about this. But so this is the GABA. This is the effect that this neuronal circuit has on the LH pulse generator. And so this GABA is inhibiting the activity of this LH pulse generator. So in order to increase the frequency, this GABA projection input needs to decrease, which could happen if this GABA is inhibiting the projecting neuronal population. I can answer more questions at the end. I just wanted to put this into context here. So what we are seeing here is that this projecting population, first as the input increases, or the stimulation increases by kisspeptin, first increases and then decreases in magnitude, which then could be associated to the effect on the LH pulse generator. Here is the interpulse interval, which decrease. That's corresponding to increase in the frequency. And this is because the output or the signal, the inhibitory signal, has decreased. So I'll show you a simulation. simulations of the whole model to kind of convince you that this is the case. So this is the second situation where we actually introduce a block of GABA cells in the MePD. And to model this, we here have to kind of weight these connections between the GABA neurons and the rest of the components in the circle. So we use this parameter beta1, which kind of represents the activity of the pharmacological block of this signaling. And here now.
So sorry, Kazi, I'm going to ask you one more question. That effect is just this antagonist is affecting the GI, but not GE's impact on the KNDy neurons?
No, because it's done in the amygdala only.
Oh, I see.
Yeah.
Applied
so
there.
Basically, the antagonist is they have this cannulae where they can selectively. Well, it's through the same surgically. It's through the same kind of tube, which is put in that region where they also direct the fiber with optogenetic stimulation, and they can also deliver these kind of. The truth is, if time goes by, it will spread in the brain.
Okay.
You go to other regions.
But I'm guessing that you're interested in a time frame, like a short time scale rather than the long time scale, right?
Yeah, we're looking at about half an hour or so. Yes, it's not.
Well, okay, so it is long. Okay.
Well, we are looking at half an hour because you have to measure also the LH pulses, and in mice they are like every 10 minutes.
Okay, I see.
Yeah.
The other question I have is the KP, the kisspeptin is constant, right? Like it's okay, you're applying it's a parameter. You're not applying some sort of pulsatile or like oscillatory signal within the KP.
No, so it is stimulated by it is stimulated by light.
No, I'm talking about the model. I'm not talking about the data.
Oh,
I'm talking
in the
about the
model
model.
is just a number. Yeah.
It's just a constant. It's just a constant. It's not an oscillatory signal. Okay,
No,
that's it.
it's not. It's not.
Okay.
Yes.
All right. Thank you.
Now we are modeling with oscillatory because for this other data that we are just having received. But yeah, here in this model that is published, it's a constant. Yes. We don't know whether it's a constant or not, but here we can think of that kind of input as the total input. Even I know that if it's dynamic, then may have more effects on the dynamics. But if you think as just like the integral under the curve or something, it's kind of a total input, which is a number. Does this make sense?
Thank you. Yes, thank you.
Yeah. So so here what we have, well, so basically here this is simulation as a busy picture, but we have the experimental data that was published in 2022 by Kevin's lab. We have now here the simulations of the model, which just came a few months ago, of this model that I just introduced, where we're coupling that neuronal circle in the amygdala and the KNDy neuronal network in the arcuate. So the timescales are very different. One of them is operates in the order of seconds and minutes, like seconds, and the other one is minutes. As I mentioned, we get pulses every 10 to 20 minutes, while in this little circuit here in the amygdala, they can be order of a few tens of seconds. So there are several orders of magnitude difference, at least 2, and what it actually results in is that limit cycle that evolves on a torus. That's not very surprising. In the paper, I'm not showing this here, but in the paper we have a bit more analysis of this and how this occurs and when and how in terms of parameter dependence. So we have basically these very fast oscillations here that we see in the silent phase, and these excursions, again, fast, that corresponds to those pulses, physiological pulses that are measured in the blood. And when we change here the parameter beta, we can see that we can reproduce this experimental experimental result where the interpulse interval increases or the frequency decreases. And we can also, like kind of study the areas or the regions in the parameter space where we have oscillations and places where we don't have oscillations, depending on the effects of various antagonists, GABA or glutamate antagonists. And we can basically reproduce all this data. That's the point of this graph. But I want to now go back to the saddle node separatrix loop bifurcations that I started with in the beginning. When we constructed the two-parameter bifurcation diagram in those two main parameters, Kp and beta-1, Kp for the kisspeptin stimulation and beta-1 for the effect of GABA suppression or GABA antagonist, we kind of found this bifurcation structure where you can see that we have this. So the purple line here is a home clinic, the yellow is a SNC, and then we have another home clinic. Here, this is just to show that there is a region which something like that I haven't seen before, but it's not something surprising. We have here oscillations in the system that are born in SNC and disappear via home clinic, or vice versa, depending. As you increase the parameters, they appear via SNC and then they terminate in a home clinic. This is what this line here is supposed to show. But we also find these transitions from home clinics to SNC and from SNC to home clinic, which basically sparked my interest in and that literature review that I gave you in the beginning. I started looking for this, and then I found all these things that I described to you in the very beginning. We were interested to find what the significance of this basically bifurcation structure might be on the behavior of the whole system. And here are just a couple of slides to show that what we are motivated by this notion of switch or a change in the dynamics. Here, this graph shows some brute force simulations where we look at the total output, similar to what I was just explaining to Anmar, that is produced by the model in terms of balance between inhibition and excitation and effect that it might have then on the pulse generator activity. So what we find is that this transition here is associated with basically positive output from the amygdala. That's the red. It's greater than 0 compared to that region here where we have blue, and we would have suppression of the activity of this population. And this is here just some examples of. So that blue and red is being computed by actually taking the area under the curve and subtracting between the red and the green here. Interestingly, here the green is the positive and the red is the negative. I should ask Kay to change these colors, but yeah, that's how she's colored them. And yeah, and then subtracting them and looking, then coloring these regions. And then if you go even closer here to these bifurcations, you can see that they may be associated. We can kind of think about a switch in the excitation inhibition balance of this output, which is quite important for not just for this particular circuit and system, but more generally for other types of brain and cognitive functions. So now, how generic are these bifurcation structures? We went to the original Wilson-Cowan model to look for this, and that was suggested by Izikevich, as I showed you in the beginning. And yeah, it wasn't very difficult to actually find very similar bifurcation, two-parameter bifurcation in the just Wilson-Cowan model. One excitatory, one inhibitory population, and this is the input that stimulates these populations. So we have this homoclinic to SNC transition, and we have the SNC to homoclinic. First, zooming on to the homoclinic to SNC transition, and just reminding you about, I like this schematic representation because it shows it quite clearly, the saddle node and the saddle colliding and becoming a SNC. And if we just look here, before and after the phase portraits, before and after, we can clearly see this in the Wilson-Cowan model. This is before, where we have a home clinic. This here, red, is very close to the transition, where the saddle node and the home clinic are kind of almost on top of each other. So that's the as close as we can get approximation. Here is the black star, where we have this this basically switching behavior, and after we have now a SNC. And initially we thought that this this here is a very similar transition. However, expecting the phase portraits actually reveals a very different picture here. What we have now is I'm just showing you how the phase portraits look before and after again. So if you look at this red star here, where we have SNC, yeah, we have that kind of SNC, the blue closed kind of orbit here. At the actual transition, what we see is this kind of connection, which I'm gonna talk a little bit more later. And after the transition, instead of something here appearing or disappearing, what we see is the saddle node is gone, and the home clinic is actually formed using this saddle node, which is very different compared to what I showed you here, where we basically have these two combining and leaving us with.
So out of curiosity, do you have a heteroclinic at least with the black star?
Yes,
here?
Yes, we do. That's exactly what I'm going. So we modified the parameters to try to get these two to combine and see what's happening at that kind of, we call it an organizing center, where this SNC appears, and it is indeed. So we call it heteroclinic bifurcation. Thank you very much, Anmar. We came up with this name, and yes, it is that kind of heteroclinic connection that appears, which now I'm gonna briefly go over some analysis that we've done, which is purely dynamical system analysis. We propose this as an organizing center. It's a heteroclinic connection between hyperbolic and non-hyperbolic, a saddle node and a saddle. And we have these global connections, gamma 1 and gamma 2. We use for kind of Poincare sections or sections to kind of study the local and global dynamics and transitions between those sections, to kind of try to characterize this heteroclinic loop. And that's just kind of quite standard, but yeah, it's not been done before, so we've done it. Kate did it. So the local map around the saddle node is the saddle node bifurcation normal form. So that's quite straightforward. You can solve these equations and you can find an explicit expression. The local map around the saddle point, we use this system of ODEs, and you can solve them again, and you can find that one too. For the global map approximations, we used polynomial approximations like Taylor expansions, and doing all this, we can basically construct return maps in any direction that we like by superimposing those expressions for these local kind of transitions that I just described. And this then allows us to kind of basically, I should have said I went too fast. We use 3 unfolding parameters. That's the minimum in order to be able to kind of fully unfold this heteroclinic loop. So here is we look at, I mean, Kate has computed this for different combinations of parameters. This is the one that we can then map back to the Wilson-Cowan model. That's why it's the one that I'm showing here. So when mu1 is 0, in mu1 and mu2, we can basically, these curves are computed explicitly based on the calculations that I just showed you, and these phase portraits describe the dynamics. Just to focus on the one that we can map back to the Wilson-Cowan model is basically the transition here at the intersection of when the organizing center, when all of these three parameters are 0, and we can see here the actual sneaker-clinic bifurcation before and after the transition, and how this maps onto the Wilson-Cowan. As I said, we are just finishing this study and we'll be submitting it soon. Something which is not in the paper that we will be submitting is, but I think is interesting, and I thought I'd show you as well. So what we also can find here is transition from SNC to SNC, which is here in the same, following exactly the same presentation. We're looking at before and after this transition. We have a SNC here, which is basically, as they call, well, in dynamics, for those of you who know, this is a non-central kind of SNC. Here is the transition, and we basically move from to a central SNC, which has, so the saddle node here is that, in this case, before, the saddle node is here, low excitation, low inhibition. In the case after transition, we have high inhibition, high excitation, and you can see what effect it has on the actual dynamics. This is important because in this case, the system overall spends longer time in the non-excited state. You have this classic kind of oscillations, action potentials. However, here you can see the system spends much longer time in the excited state. So it is, it's potentially of interest to neurodynamics. Still figuring this out anyway. So in summary, we have proposed and analyzed a model of this amygdala circuitry that could serve as a basis for future modeling of the effects of stress on endocrine functions that are controlled by hypothalamic nuclei. We investigate the saddle node Separatrix-Loops bifurcations detected in the model, along with associated implications for neuronal network dynamics. We identify and unfold the type of saddle node Separatrix-Loop, which we name sneaker-clinic, and our analysis enables us to investigate the role and significance of the saddle node Separatrix-Loop bifurcations in controlling the balance between excitation and inhibition in the system. And before we move to questions, I did mention future work. Basically, now what we have is activity recorded using green lens. This is calcium, actually, dynamics or calcium transients. I wouldn't call them spikes. Basic calcium fluctuations in these neurons. These are GABA neurons in the amygdala, in that same region. And we are analyzing this activity and linking it at the same time. In Kevin's lab, they also can record the activity of candy neurons using the same kind of green lens technology. This allows you to record individual cells, but also populations. This is just here an example of population activity in the arcuate nucleus, but we also have individual neurons there as well, all done in live animals. And we are modeling some of the effects now of stress, trying to connect the recordings in the amygdala to the recordings in the arcuate nucleus. But that's very much work in progress. It's literally coming out of the lab as we speak, that these recordings, so we're very early days with these ones. Thank you very much for your attention.
Great, thank you so much. So time for questions.
Yes. Shall I stop? Yeah, I should have acknowledged people. That's it. These are all the people
Yes,
that.
of course. Please go ahead.
Yes. So shall I stop sharing now?
Yes, you can stop sharing. But maybe people have questions and they want you to go back. So
Yeah, let's. I'll keep it for a while and
yes,
then.
yeah.
Can I ask a question?
Yes,
Yes.
of course.
Actually, it's rather a comment rather than a question. So I really like this terminology is sneakocolic, sneako,
Clinic.
whatever. Yeah, snake with homoclinic, combining it.
Yes.
I just want to mention one thing. There's an immune system model that was published a few years back that is essentially has the same sort of features. These transition from a SNCC to homoclinic and homoclinic to SNCC. I would recommend just linking it to that study. You generate these. The model also generates these heteroclinic orbits as well that
Yeah, so
combines a homoclinic with a SNCC as well.
is it a model of development or something or cell cycle?
It's if I'm not mistaken, it's a T-cell model. I'm gonna dig it up right now, and I can send it to you
that
through the
was
chat.
useful because I found one very recent one, I think last year, from the cell cycle kind of.
No, it's not a cell cycle. It's an immune system model. It was published, I would say, 6 or 7 years ago. I'll dig it up right now, and I'll send it to you.
I don't know about that one because, yeah, as we have observed this in our model and then look into the literature, and first it was about neurons, but then we are also finding it in other models. Like, as I said, there is this. It's actually from this group in Vancouver that is looking at development modeling and cell cycle. And
I see.
Eric, I can't pronounce his second name. Do you know who I mean?
Eric Citrenbaum. Eric Citrenbaum.
Yes.
Yeah.
They have it in one of their models, a paper that appeared
As
last
well.
year.
Oh, I see.
Yeah.
But like I said, this is the features of that model very, very similar to what you're seeing as well in your model. So I'm gonna forward it to you shortly.
Thank you, Anwar. That's very, very useful because, yeah, the more we, I think it's very fundamental. It's just it just hasn't been previously looked at really in detail.
More questions
from the audience?
Yeah, I could ask a question.
please.
So I'm not clear of what features in the experimental data indicate that these sorts of bifurcations are appropriate to model the data.
So I guess the only well, okay, so it's because they're models of neuronal dynamics, whether it's a single neuron dynamics or like the Morse-Lecard, for example, or it's Wilson-Cowan, which is about neuronal populations. It's that kind of rationale here behind it's they kind of appear indeed. It's not, we are not using the bifurcations to model the data. We find the bifurcations in our models basically, and then relate them to behavior, which might be physiologically important.
But I think that in the SNCC bifurcation, one of the characteristics is as a parameter goes through a critical value, you start with an oscillation that's initially very slow and then gets faster, but always at the same amplitude. And that distinguishes it from a Hopf bifurcation. So your experimental slides, to me, were very interesting. And I was trying to see if there's some aspect of what you see, because we can, you can make up any model and find all kinds of bifurcations. Just because it's a model that everybody uses doesn't mean that we should believe it. But
no, absolutely.
In the data, I was just unclear exactly what kinds of experiments people are doing in the data that indicate this class of bifurcation is happening. Is there some evidence in the data that there's a SNC bifurcation, for example?
So in our data, because it's so there are several things. We are just starting to get a glimpse of that population data dynamics of these brain regions that I was talking about. And I showed you some, but we haven't really analyzed. First of all, it's calcium. It's a calcium dynamics. It's not like electrical activity. So, but having said that, it doesn't mean we won't find it, or we definitely need to do what you're suggesting, Leon, in terms of looking at this, whether the amplitude changes or doesn't change, so that we can associate it with this, with the SNCs or HOPs or home clinics and things like that. But yeah, but we haven't done that, and we should be doing this as we get more data, which is currently being collected. In the paper from the German group, they actually found, but it's not a SNC. Over there is a home clinic. They have the home clinic dynamics in there, but it's a patch clamp recording there. It's much less noisy, and it's much more reproducible, if I can put it that way, because you're just looking at a single cell in a slice. In our data, it comes from in vivo experiments. It's very noisy, it's very variable. We need more data in order to be able to kind of more conclusively say something.
In one of your slides, you showed there were several traces, and on the right side of the slide was when you did some procedure, maybe change the activity of the light. And I thought that the left-hand side was the control. Is it? Do you know which slide that is?
Yeah, I'm just trying to get...
What?
This one?
No. It was before that.
So before that, I was showing slides. So all these ones here.
An experimental slide.
Yeah, it's coming. So here, all these ones are actually hormone levels in the blood. So this is quite...
Yeah.
But then I showed the slide from the...
I think that's it. So
yeah,
on the left, in the, like on the right-hand side where the experimental records are, but the leftmost column.
Yes, here.
Yeah. Is all of that before the black bar the control?
Yes.
So the controls look quite different from one another.
Yes, it's very variable because it's a live animal.
No, I'm very, I like that, but the fact that it's very, is it meant to be? It's. Can you just say a little bit about the animal?
So, okay.
Mouse, or what is that animal?
It's a mouse, and basically this is just a representative trace. If you see here, the way these things get quantified is by looking at the frequency or the interpulse interval, which is here on the...
No, but I look at everything,
Yeah, I know, and you're right to look at everything. But now what we're starting to get is actually, see, these are model simulations, but now we're starting to get these kind of thing from the actual neuronal populations. That was the last slide, where we see much more reproducibility, if I can put it that way. Well, I mean, these are also reproducible in terms of frequency, in terms of shape, in terms of amplitude. They are very variable because different mice, the assays, this is like basically ELISA, where you probably know, you have a standard curve and you have to relate the amount of hormone to standardize it, and then things can look very different. Yeah, it's quite.
Are those female mice or male mice or both?
These are female mice. Female mice, yes.
Female mice.
Oh, and also it depends on the stage in the cycle as well. So we are doing them not in like most experiments. Most traditionally are done in so-called OVX mice, where they over-optimize the mouse, so it doesn't have these feedback effects from estrogen onto the activity. But my collaborator does this in intact mice, basically no surgery, but that makes it even more challenging.
So wait, are these are these having the estrous cycle, or they're not? They are.
So this is the estrus usually, because that's when they have, in estrus, they don't have any pulses. They're basically silent.
So this is on the same day
Yeah, yeah.
and the same time of the day, do you think? Or were they controlling that?
I'm not sure, because it's quite, well, they at least control for that day, because they use they have markers to find out whether which day of the cycle the animal is. So when they do the experiments in a specific day on the cycle, they control for the day. That's right. But then for the hour or something like that, it's hard specifically.
Okay, I mean,
But these are very valid questions.
the data is really interesting.
Yeah.
Thank you.
More questions?
There was something in the chat, maybe, or not really?
Just Anmar sending you the link to the paper from 1995 chaos.
Okay, great. So maybe I can somehow see the chat.
Okay, there are no other questions. Then if you've taken down the link, if you've clicked on it, if you have the link, then we can.
That's what I'm doing right now.
Yeah.
Yes, not yet.
So if there are no other questions, then yeah, we can say thank you very much to everybody. And thanks, Krasi, especially for a great talk and for the questions. And see you next week at the next seminar. Thank you very much. Okay.
Thanks,
Bye.
Krasi. I really enjoyed your talk. Okay.
Thank you
See you
so much.
next year.